Hello again, dear students. I hope that you're doing well. Our lesson today is about human karyotype. The objectives of this lesson are to define a karyotype, to identify some uses of karyotyping, to determine the criteria used to arrange a karyotype, and finally to write a chromosomal formula. Now let's start. As an introduction, a karyotype is simply a picture of a person's overall number of chromosomes. As I explained in the previous video, chromosomes are located inside the nucleus and they are carriers of genetic information. In order to perform a karyotype, first, chromosomes must be isolated, most often from white blood cells nuclei. Then they will be stained or dyed with certain colors. And finally, they will be examined under a microscope where chromosomes are identified and photographed, and then they are cut out into pictures and arranged into a karyotype. Chromosomes are not arranged randomly. There are certain criteria which control arranging them into a karyotype. Now this image shows how chromosomes look like when observed under a microscope before being arranged. Here, G-banding technique is used to stain these chromosomes. After being arranged correctly, we obtain this karyotype. First, you can observe that they are arranged in decreasing size. It means that they are arranged from taller into shorter ones. And they are arranged by similar banding patterns, which are these white and gray bands, and also by same position of centromere, as in here. Moreover, you can observe that they are arranged in pairs called homologous. So chromosomes are arranged in pairs which are called homologous, and this term will be explained in the next slide. So what are homologous chromosomes? This schema shows two pairs of homologous chromosomes which are made up of chromosome pairs of the same length, same position of centromere, and similar staining or banding patterns. So what is a band? A band is defined as the part of a chromosome that is clearly distinguished from its adjacent segments by appearing darker or lighter in color. So dark bands must be aligned at the same level as dark ones, and the same goes for light bands. Now, this pair of chromosomes is non-homologous since they are of different length clearly. So to sum up, the criteria used to arrange chromosomes in a karyotype are: first, decreasing size of chromosomes, then similar or same banding or staining patterns, and finally the same position of centromere. So to define a karyotype clearly, we will say that it is a representation of all chromosomes of a cell in order of size and shape. Now we have two karyotypes, which are karyotype 1 and karyotype 2. You can notice that these karyotypes have the same number of chromosomes, but chromosomes of karyotype 1 consist of two sister chromatids, while chromosomes of karyotype 2 consist of one chromatid. But what are the benefits of karyotyping? Karyotyping allows your doctor to determine whether there are any abnormalities or structural problems within your chromosomes, which will be explained in the next video. We've mentioned earlier that chromosomes are arranged in decreasing size in a karyotype. But how many chromosomes do we have? And do all of us have the same number of chromosomes? I'm sure that these questions are roaming your heads. First, not all living beings have the same number of chromosomes. So number of chromosomes varies among living beings. For example, a pea plant has 14 chromosomes while sunflower has 34 chromosomes. A dog has 78 chromosomes while a cat has 38 chromosomes. But among individuals of same species, number of chromosomes is constant. It means in dogs, for example, all dogs will have 78 chromosomes. Whereas for humans, we have 46 chromosomes or 23 pairs of homologous chromosomes. Now let's move to types of chromosomes. Chromosomes are divided into two types, into sex chromosomes, also called gonosomes, and into autosomes. Gonosomes or sex chromosomes are the pair of chromosomes that determines a person's gender. They are of two types, X and Y. X is the longer one, while Y is the shorter one. Gonosomes are found at the right corner end of a karyotype, here, while the other pairs from numbers 1 to 22 are called autosomes. So how to determine gender using karyotype? If a karyotype has X chromosome and Y chromosome, so this karyotype belongs to a male, and if it has X chromosome along with another X chromosome, so it belongs to a female. Now let's move to an important part of our lesson, which is chromosomal formula. It's important since it allows you to answer several questions which are asked in official exams. So a chromosomal formula states the total number of chromosomes, which is 46 or 23 pairs in case of humans, or the total number of autosomes, which is 44 or 22 pairs in case of humans also, along with sex chromosomes, which are XX in case of female and XY in case of a male. It also states if there is any extra or missing autosomal or gonosomal chromosome. Now let's do an application which will help you to understand how to write a chromosomal formula. You can find it in your book page 187. Here we have two karyotypes. The first one is for a woman and has XX gonosomes. The second one is for a male and has XY chromosomes. So we have to complete the document referring to the above karyotypes. Let's start with the woman. So the number of autosomes in this female is 22 pairs or 44 chromosomes. The number of sex chromosomes is 2. The sex chromosomes are XX. So the chromosomal formula will be written as total number of autosomes plus XX, which are the gonosomes, or the total number of all chromosomes, which is 46, comma, XX. Now, in case of the man, we have 22 pairs or 44 autosomes. The number of sex chromosomes is also 2. Gonosomes in case of the man is or are XY. So the chromosomal formula will be 44 plus XY or 46, comma, XY. A chromosomal formula can be written in another way, which is 2n equals total number of autosomes, which is 44 in case of humans, plus gonosomes, which can be XX for a female or XY for a male. So n is the number of chromosomes in a cell's nucleus, whereas 2n represents two sets of chromosomes, since chromosomes are present in homologous pairs. So in a human diploid cell, the chromosomal formula will be 2n equals 46, which is the total number of chromosomes, while in human haploid cells, the total number of chromosomes is 23, so n will be equal to 23. Diploid cells and haploid cells will be explained in the next video. So now the chromosomal formula for a human male is 2n equals 44 plus XY, and in case of a human female, it is 2n equals 44 plus XX. Finally, this is an application about arranging chromosomes into a karyotype. We have 8 dispersed chromosomes which must be arranged into a karyotype according to decreasing size from tallest to shortest and to same position of centromere and similar banding pattern. We start with the tallest homologous chromosomes, which are paired according to same length, same position of centromeres, and similar banding pattern. We continue arranging the other pairs in decreasing size. The remaining pair is non-homologous since it consists of X and Y chromosomes, X being the longer one. So we have pairs 1, 2, and 3, and X and Y chromosomes. Pairs 1, 2, and 3 are called autosomes, while X and Y chromosomes are the sex chromosomes. To write the chromosomal formula for this karyotype, we can say 2n equals 8, which is the total number of chromosomes, and we use 2n since we have two sets of chromosomes, or we say 8, XY. We can also use 2n equals 6 plus xy. 6 is the total number of autosomes, or simply we can say 6 plus xy. We usually use these two formulas. This karyotype belongs to a male since we have X and Y chromosomes as gonosomes. Thanks a lot for your attention. If you have any question, don't hesitate to contact me, and please stay home and stay safe.